Okay, good morning. Let's have a class. We're gonna move to the exam 3. We'll talk about biochemistry and the genetics. So exam 3, we have roughly 7 lectures, including a guest speaker in November 6th. So I'm not gonna be here, and Dr. Scott Baldridge will give a talk about the parasite. It's a scheduled guest lecture. But other than that, we are going to talk about this biochemistry. So we'll have a biochemistry in 2 classes and plus genetics. If you are currently taking genetic classes or you are taking biochemistry classes, all kinds of biochemistry classes, which certainly helps. But keep in mind, when you guys are talking about is eukaryotes, so human beings, animals, plant cells, we'll talk about the bacteria, so there is a little bit of difference. For the biochemistry in here, we are focusing more is ATP generated, the energy created, how we do the calculations, that will be the key parts. Also, during here the section, there is some basic knowledge. I think I need to help you to fix it, because although you took a couple of classes or different classes, I don't think people understand those things very well. So I try my best to fix that and let you understand it. Genetics, we're going to do the DNA, RNA, then go to the protein. Very similar to central dogma, we're going to talk about DNA replication, transcription, and translation. And if we have time, we're going to talk about the lacto-open. So those are basically is outline what we have. It's going to have lots of chemistry. We're going to talk about that. And you're going to see me draw all the blackboard, heavy chemistry in the next 5 or 6 classes. So you have to do a little bit of review, and I also going to mention, we'll offer you very heavy extra points during the exam 3. Because after exam 2, you will see some people doesn't do very well. Some people do very good. Like I said earlier in the lab 1, exam 2, you should give me your exam paper in 15 minutes. Then you're solid A. If you stay there for like more than 1 hour, it's not going to be very good, because the more you're reading, the more you're confusing. That's what happened. So it's so many bacteria strain names there. If you never heard about that, it will be difficult. But exam 3, we try to help you. There is at least 30 or 40% of the students in the classroom that need some help. So we help you to get as many as you can. So we offer lots of the extra points, but you have to put some work there. So I will talk to you later on what we're going to do, okay? So we talk about biochemistry. Today is an introduction. First of all, we talk metabolism. Once we talk about metabolism, you always should know there is 2 different categories, which is catabolism and anabolism. Catabolism basically is, you look at the slides, is you have a large molecular breakdown, become a small molecular, and created ATP. And the anabolism basically is small molecular using ATP to generate a macromolecular. So this is ATP is done, is using the ATP. Bacteria has to survive in their lifetime. This has to be coagulated, or we say coupled, all the time during their life cycle. If there is a 1 second stop, the bacteria will be died. So you always remember they are connected. In this class, because of time limited, we only going to focus on catabolism. Anabolism, we're not going to talk too much. Usually, if one student is biochemistry or microbiology major, there is a separate class talk about anabolism, which is called microbiology physiology or microphysiology classes. That is usually for microbiology major students under biology department. So we're not going to spend even a second to talk about this. We always going to focus on is catabolism. Okay? So that's the overview what we're going to talk. And the definition here you can see what is called catabolism, what is called anabolism. I'm not going to repeat it. And this is the slides showing you what is catabolism and anabolism in a very layman expressed language. So you can see the energy source glucose become waste products, and they use the energy using some precursor chemicals. It's going through anabolism to manufacture the cell structures. So this is very easy to understand. So next we're going to go to the topic. Thermodynamic. So that's thermodynamic theory. This concept is not very easy to understand, although you have been learning in different classes. The idea is around 100 or 200 years ago, people want to create some machine, can working all the time. Using energy, created energy never stopped. But from the physics standpoint, it's not going to work. Then at the same time, the biology science is starting to develop, same as microbiology. So the scientists, they try to using thermodynamic, which is a physics concept, to explain biology or microbiology phenomenon. This makes it a little bit tricky and difficult to understand. So that's why we wanted to spend in time to explain to you here what this means. And I maybe jump a couple of slides, but they all covered in here. So thermodynamics we'll talk about is first law. So first law in the physics book, textbook, is not described like that. It's much more complicated. In a closed vessel, we had energy comes out. We using the heat, then the heat generated. There's a lot of the formula, even the calculus equation about that. But from the microbiology standpoint or biology standpoint, we talk it very simple. First law is energy never created or destroyed, only transferred from various formats. So said very simple, a very easy example is potential energy become kinetic energy, then the bacteria can use it. Now, I don't know whether you do these physics puzzle. In your physics class, I did a lot when we were young. So, let's say very simple. I had a ball here. This is a little bit high there. Okay, there is of course a potential energy. This is potential energy equals mgh. And then once the ball is going down, this potential energy going to move this car to moving, does become a kinetic energy, then becomes this formula, and those equal. So what happened is V equals. So that's physics. But we're not going to talk about that. Just an example. First law is energy is transferred between the different formats for the bacteria or microbiology to using. But they were never created or destroyed. So that is the first law. Okay, what's the second law? Second law, you can read. It's very complicated sometimes. That's like a one book chapter. But if we said very simple, is disorder increase? This can very simply understand why the animals will die, or human being will die, or bacteria will die. Because in their systems, the disorder is increased. And every time the disorder, they want to put them back. However, they cannot balance that. Then the bacteria going to die. So second law of thermodynamics, if we want to say it very simple, is a disorder increase. Now, in order to explain the thermodynamic, it comes out a very important equation, which is called the Gibbs equation, or Gibbs principle, which related to delta G equals delta H minus T delta S. So what do these things mean? Delta G is free energy amount. You can easily understand it. This is the energy used for doing work, which means the bacteria can survive. That's a delta G. And what is delta H? Delta H is called enthalpy. That is amount of heat content or change or say variation in a fixed pressure and volume. So go back to here to the thermodynamic. This originally, what we talk about, is a closed vessel. The closed vessel has a fixed pressure, has a fixed volume, and also could be impacted with temperature. So in the physics area, you have all these type of things comes out. I don't know if you're familiar with that. The heat balance equation. Those are originally how they describe or originated from the thermodynamic. So delta H is enthalpy. The amount of the heat contents change in a fixed or variable pressure and volume. Okay, what is T? This is temperature. Everybody knows. But be careful, the T here, it is not degrees Celsius. It is not degree Fahrenheit. That is the international unit of the temperature is Kelvin. That equals degrees Celsius at 273. That's called Kelvin. It's a little bit different. What is delta S? Entropy. That means amount of the energy cannot work. So these are the definitions. Now we're using this Gibbs equation to explain how the reaction will be happen, how the biochemical reaction will happen, and the chemistry reaction will happen. And you already know the conclusion. If delta G less than 0, spontaneous reaction will be happening. If delta G more than 0, the spontaneous reaction will not happen. If delta G equals 0, it's an equilibrium status. So you already know the conclusion. But how we get that, what that means, I want to explain to you very simple here. This is the conclusion. Every textbook have it, and that's it. So what's the story behind it? So let's talk about this equation. We want to explain this to you a little bit more. So let's talk about delta H. From here, if we talk about delta H, then we assuming T and delta S are not going to change. Okay, if delta H decreased, let's say original number is 100, right now become 90. Of course, delta G got to decrease, they're going to become 0. That means spontaneous reaction will happen. The example is, I press a ball, go down. The ball up here and compared to the ball in here, the delta H is decreased. That's why the spontaneous reaction will happen. That's the first explanation. How about the T? Let's assuming delta H and delta S all stay the same. Once we increase in the temperature, the biochemical reaction could spontaneously happen, which means it will happen. What's the explanation for that? Cherry bomb. July 4th, firework. You got a lightning, a cherry bomb. The temperature is increased, then boom, it's going to go up. That is temperature increased. And a spontaneous reaction will happen. Okay, what is delta S? Delta S is relatively easy. If I have a closed box, I have these small balls here. This is a very organized status. If one day this block is gone, and then this ball goes here, that's this organized. From here to here, delta S is increased. So spontaneous reaction will happen. Assuming delta H and T is not going to change. So that's from a physics standpoint, how we explain this one. Now, in a microbiology or biology standpoint, we have a very standard reaction. Can explain it. And that is using a very universal formula. Chemical N and B will generate chemical C and D. So therefore, we have an equation for that and the constant for that. So remember, when we talk about the biochemistry reaction in our microbiology life, they are all reversible. Or we say 99.9, they are all reversible. Any of the organic reactions, they are all reversible. So we get K equilibrium constant equals the product concentration and the substrate. And then we also have a very important formula for that is delta G equals minus 0.303 RT and log 10 K equilibrium. And this is a fixed formula using in the biochemical reaction. What is R? 1.98 cal per mole per degree. What is the T? T is Kelvin. Now from here you could see, because we have a log 10 here. So if this is more than 1, what will happen? Delta G less than 0. If less than 1, delta G more than 0 equals 1, delta G equals 0. So I want to explain here. Log 10, if K equal more than 1, let's say 10, what is this value? That is 1. We have a negative at the beginning. So this part is positive, you have a negative at the beginning, this value is negative. Just give you an example. So once delta G is negative, what it means? Spontaneous reaction will happen. It's called exogonic reaction. And this exogonic reaction from the energy equation standpoint, explains catabolism. So think about, there's a glucose in the bacterial cell, bacteria can use it. That's spontaneous reaction. Let's say if this guy equals 0.5, that means 10x equals 0.5. That equals the x is less than 0. That's physics. So once this area is negative, you have a negative here. So negative and negative is positive. Therefore, delta G is positive. Once delta G is positive, spontaneous reaction will not happen. That is called endogenic reaction. And this represents anabolism. Does anabolism spontaneously react? Of course not. If we want to build up something, you need energy. Think about at your home when you were very naughty in the kindergarten. You have the toy house built up. Your neighbor come here, just push it, it's gone. That's spontaneous reaction. But then you second day you're crying and you have to spend it the whole day. Your parents help you to rebuild it. Is that spontaneously happen? Of course not. That's not lots of energy. So that's explained by the anabolism. And this is equilibrium status. Basically, nothing will happen. Okay, but this is, to be honest, in the microbiology area, this doesn't happen. Okay, so usually we are talking about is right here. So that's what we explain to you, the thermodynamic. So when you go back to look at the slides, this tells you the free energy and the reaction, and the positive, negative. This tells you each index, what this means. That's equilibrium equation, equilibrium constant, and this is what I just draw there. And you can see right here, which is we talk about is exogonic reaction, endogonic reaction. Okay, next we want to talk about the energy currency. So this is the first thing. We explain to you the Gibbs equation, talk about from chemistry and the physics standpoint, explain microbiology phenomenon or biology phenomenon. It's a multidisciplinary explanation. So next one, we want to talk about the energy currency, which is ATP, and we also want to talk about how the energy going to be generated. I'm going through the two steps, and everybody knows, which is substrate level phosphorylation and oxidative phosphorylation. So the first thing we want to talk is ATP. So first the question is very simple. What is the full name of ATP? So you can ask yourself, 5' adenosine triphosphate. That is ATP. What we call it? Energy currency. But at the same time, I want to be emphasized, I want to talk about right now, not only ATP. Remember, you have GTP, you have CTP, you have UTP. These are TTP. These are could be the backup energy currency to be using in some of the circumstances. But we still going to talk about the ATP as a standard energy currency. Second, what is ATP structure? What it looks like? It's not difficult. First of all, if you do not know about these things, you just draw like this. It's a base, it's a ribose. Where the ribose comes from? Comes from pentose phosphate. Generate a ribose. And then in here, you put a base. And the base, since it is ATP, of course it's adenine, so it's A. And then how about here? It's very important that you're going to have this comes out. What's that? polyphosphate. But we are in a university. Some of you are even go to medical school very soon. This is a high school or middle school requirements. You're not going to be just that simple. But I just want to give you a basic knowledge of what it looks like. So what's a structure looks like? We usually draw from here. And you should know this very familiar. Is that correct? Okay. So how you label? 1, 2, 3, 4, 5, 6, 7, 8, 9. That's adenine. This goes here. And then you add, I didn't draw very well here. You add a ribose. Okay, of course this is not that long. What's a ribose looks like? H, OH, that's 1 prime, 2 prime, H, OH, 3 prime, 4 prime, CH2, that's 5 prime. What we're missing? Phosphates. Is that right? And then, right here. So what this says, this one called? That's tripolyphosphates. That's ATP. What is section here? ADP. What is section only here? AMP. That's what it is. So what's the extra points in the exam 3? 5 points to draw the ATP structure. That's not difficult. You already learned in your different classes. The only difficult part is right here, the base. We are talking about the DNA. We're going to mention that again. Okay, that's ATP structure. The formula is very simple. You know ADP, other phosphates become ATP. If we want to talk about the energy, this is called spontaneously happening. And all the other, it's called spontaneous happen during the catabolism process. What is the delta G? Minus 30.5. That's what the ATP created, how many energies they needed. Now, the bacteria is very smart. During their, we call it's energy biochemical generated cycles. They have some chemicals has much lower delta G than this. And at this moment, the delta G, we put absolutely value. Become phosphate transfer potential. And the number to evaluate how much capability of the chemicals can transfer phosphate. And the phosphate is very important. And the people find that there are some chemicals has much higher absolutely value of minus delta G, which means they have a much higher phosphate transfer potential than ATP itself. For example, phosphate enolpyruvate. This is minus 61.9. That equals absolutely value 60.9. Glyceroldehyde 3 phosphates equals 49.5. What this means, the transferring capability could be more than the ATP generated by themselves, so they could transfer in phosphates and become ATP. This is called substrate level phosphorylation. That's what it is. Now, where is phosphoenolpyruvate? Remember glucose? Very bottom here. Finally is pyruvate. The second to last one is phosphoenolpyruvate. What is that? That's phosphoenolpyruvate. It's right there. Where is glyceroldehyde 3-phosphate? Also, sorry, 1,3-biphosphoglycerol. 1,3-biphosphoglycerol. This one made a mistake here. Phosphoglycerate. Where it is? So right here. What on it looks like? One 3-biphosphoglycerate. Why we saw it's a glycerate? That's aldose, the simplest sugar. And you add a phosphate. Is that right? So that's very basic chemistry. So that's why they can do substrate level phosphorylation, because the energy transfer potential of phosphate is larger than the ATP itself. But then people will ask some questions. During the glycolysis process, you have some chemistry right here. Let's say glucose 6-phosphate. You also have a phosphate. Why you cannot do it? Glucose 6-phosphate. Remember how we do it? We draw glucose. That's glucose 6-phosphate. Why we cannot do it? Looks like complicated. You draw it like 30 seconds. Why you cannot do it? Because this delta G roughly only 13.1. The delta G value is less than that. It's not enough to combat the ATP generated by itself. So you cannot do substrate level phosphorylation. Same as glucose 1-phosphate, which is using during the translocation. So just want to let you know, that's even less, only 9.2. And that's a reason why they cannot participate the substrate level phosphorylation. Okay, so lots of the time we had a question in the biochemistry. What is the substrate level phosphorylation? Lots of people, lots of the teacher will teach you substrate level phosphorylation is not using electron transport chain. I don't think it's a very good definition. They just say it's not an apple because it's orange. Or in the older time we say it's not a boy because it's a girl. We no longer can say that. That's not a definition. And this is a good definition. Which means from the energy standpoint, because the phosphor transfer potential, taking the absolute value, is larger than 30.5. So that's why those chemicals can do the substrate level phosphorylation. Okay, so that is a definition for the substrate level phosphorylation. Okay, next. We want to talk about another important concept, which is called redox reaction. That's oxidizing and reduce equation. And also related to the electron transport chain. And I want to talk about here because it is certainly connected to what we did in the lab for the oxidase test. Because oxidase test is related to the electron transport chain. So we want to talk about that in a little bit of detail. Okay, first of all, redox reaction. That is reduction, oxidizing, or we say oxidating, the oxidation reaction. So we want to review very quickly in the chemistry. Chemical A and B, in a reversible reaction, generate chemical C and D, and A transfer electron to the C. What are these things called? Okay, what is C called? And I tell you a very simple way to understanding. I don't know whether you're still doing this exam. When we, 20 years ago or 25 years ago, when I applied the university in here, as an undergraduate, we need to take a class called the GRE test. You heard about that. Go to the graduate school. It's not required. Lots of universities does not require that anymore because of the COVID-19 pandemic. So gain electron means reduction. So how you understand about it? Think about the 18 years old, the girl's face. Looks beautiful, a lot of energy, lots of electron. So you gain the electron, it's a reduction status. So when you hear what's C? Reducing product. Okay, the energy transfer, the electron transfer has to be balanced. If you transfer to here, of course, D losing something. So what is D called? Oxidation product. Then what is A called? That's called oxidizing reagent. Then what is B called? Reducing reagent. That's a very basic chemical reaction. Now what means oxidation? What means oxidize? Lose electron. Think about 90-year-old grandma's face. That's not much energy there. That is oxidized. And the bacteria have to survive. Their reduction and oxidation has to be coupled. Which means the electron transfer has to be circulated and always do that in every second. If for some reason they stopped, the bacteria will going to die. So this electron transfer here is very important. Okay. Now how are we gonna express that to you? This is something we want to talk. Electron, we're using delta E to express. Electron transfer, they always have electron donor, and they always have an electron acceptor. And the electron transfer always from donor to acceptor, and you know about that. In the physics standpoint, they can measure them using delta E0 prime, and they could be measuring use volts or voltage value. And this voltage value, which means index the electron transfer potential. If the value more negative it is more potential an electron donor. If the value more positive, which means it's more potential an electron acceptor. That's what it is. They use the voltage value to measure that. Now, we talk about delta G all the time. What is the delta E and the delta G is a relationship? Delta G equals minus delta E and then F. This is called Faraday constant. And that is N, and that is approximately 10 to 3,082 kJ per mole per degree. So what this tells you, we're using voltage value to mean the electron transfer potential. At the end of the day, it is still connected to the delta G. So it will still go back to delta G positive or negative. But we don't want to get to that, because if we get to that, it's completely become a analytical physics or chemistry. So we're using another way to talk. Okay, how are we going to do that? Then we go back, talk about electron transport chain. Which way is a good one? Okay, that one is good. So let's talk about the electron transport chain. ETC, electron transport chain. When we talk about that, we're going to go back to mention here. Okay, electron transport chain. Everybody knows it's very important. First thing, what are the location? It is different. For bacteria, because it is a prokaryotes. So where is the electron transport chain? Cell membrane system or cytoplasma system, whatever they call it, it's a cell membrane system. How about fungi? This is same as you learned in your genetics or biochemistry class. It is in a special structure called mitochondrion. Okay, it's a special structure. Second, what is the electron transport chain looks like? A standard one is NADH2, go to FAD, go to FMN, FEX complex, and go to coenzyme Q, go to cytochroma C, go to cytochroma A, then go to cytochroma A3, and the primary goes to oxygen. That's a standard electron transport chain. Now we have some questions we want to solve. First of all, why the order is like this? There's a reason for that we just talked about. The more negative voltage value is more potential electron donor. The more positive value is more electron acceptor. That's the first thing. And this is, we also call it two-half reaction. And why we say two-half reaction? Because half of that are reduction, half of that are oxidation. So let's say NAD. Let's say this one. What is this called? Nicotinic adenine dinucleotides. Okay, what's this one? What's the voltage value? Very negative. Minus 0.32. Okay, how about this? What's the voltage value? Positive 0.82. What's the distance between that? Roughly 1.14. This is the big difference between here to here. Then why FAD is here? What is FAD? Fulving adenine nucleotides. What's the FAD value? You can look at here. 0.2. That's less than minus 0.32. That's minus 0.2. What is FES? You look at the table. That is about 0. They start from coenzyme Q, cytochrome C, A3, and oxygen. It's all become positive. So it's all potentially to be an electron acceptor. And the final electron acceptor is oxygen. Why we do that? Because we think this is a big distance from here to here. The big voltage value distance indicates a more energy going to be generated. Okay, so that is a basic knowledge of what's the electron transport chain. Now, related to the concept of what we said, substrate level phosphorylation. What is oxidative phosphorylation? Oxidative phosphorylation, there are at least 3 or 5 theories. Lots of them we're not going to talk because we are microbiology class. We want to talk a very important one is in 1932, Peter Mitchell, a British microbiologist, a physician scientist, who is generated a theory called chemosmosis. This is the explanation, very good, to line up what we talk about is potential energy. So he thinks about electron transport chain, let's say, in the bacterial cell membrane system. During the electron transport chain, transfer electron, there's something going to go out of the cell membrane system. What are they? You have positive stuff transferring, you of course, or negative stuff transferring, you of course have some positive stuff going on. We call it a proton. The proton is transferred from the inside of the cell membrane, go to the outside, and accumulate. So all these things accumulated there. This becomes a potential energy. And we just said about energy will never generate or created, it's only going to transfer. Once you have all the potential energy generated there, what this means? If there are so many things outside, you're going to have to keep the balance. They have to go back. So, these protons, they could come back. And once they comes back, what they do? They activate an enzyme called ATPase. So once you see ATPase, what this means? Of course, it's that ADP with phosphates generates ATP, and that is relying on an enzyme called ATPase. It's a halo enzyme, multi-complex halo enzyme. Has a core enzyme and the protein components, non-protein components. We'll talk real quick about that. So the ATP generated. Okay, once the ATP generates, what are we going to do? Of course, the nutrition could come in. Once the nutrition comes in, how do we know there is a kinetic energy? Expressed in a bacteria, flagella rotation. Usually around here. That is the reason why ATP could generate. Because of the ATPase has been activated and triggered by what? The electron transport chain to push the proton, created a force. What is that force? Proton motive force. So this is also called PMF theory. That's a proton motive force. This is a good explanation. What is we call it? Oxidative phosphorylation. And right there. That's oxidative phosphorylation. And with the electron transfer, the proton originally is inside of the cell membrane system. They pushed out. Now, of course, when they push out, they need the enzyme. What's the enzyme they call? To take proton outside, which means you take a hydrogen comes out. What is called? Dehydrogenase. It's very good terminology for that. So that's why we have NADH dehydrogenase to do that. So once the proton has been accumulated outside of the cell membrane, it becomes a potential energy. A bacteria cell always like that, or biology products always like that. Too many is there. They are trying to come back. And once they come back, the ATP synthesis, or we say ATPase, is being activated and triggered with active transport. That's what we learned. And then coupled with flagellar rotation. And this is how they generate ATP. Now, how much ATP they going to generate? Remember, NADH go to oxygen. That is 3 ATPs. This is when I was student. Right now, what do you call it? 2.5. Because you are thinking about oxygen phosphorylation. How many ATP from FAD to oxygen? When I was student, it was 2 ATPs. Right now, 1.5 ATPs. Now, why NADH created more ATP than FAD? Because the distance here is more than the distance of FAD to the oxygen. That is go back to see the electron transfer potential, the voltage value. Okay, so how that works is all connected with each other. So these couple of slides, I have been conclude them, which is talk about reaction, oxidation reduction, two-half reaction. What is that? This is some examples, the location, and this is important when we talk about. This table here talk about the electron carrier, and we talk about NAD, FAD. There is no one we have mentioned is NADP. This NADP using in biosynthesis, for example, Calvin cycle. And we're not going to mention because that's anabolism. Okay? So that's an important slide. And this is important slides because talk about chemosmosis, Peter Mitchell in 1932. This single slide is a Nobel Prize. Biology Nobel Prize in 1940s-ish. Because usually they delay a little bit. And this explains the substrate level for oxidative phosphorylation. But this is only one theory. Why we want to talk about here? Because it's related to the bacteria, related to the rotation of the flagella. Okay. Okay, metabolic pathway. We know the metabolic pathway have several things. It could be linear, it could be branch, or it could be circular. We have 3 very good examples. That is what we're going to talk next week, Tuesday. We talk about energy created, destroyed, not destroyed, created, transformed. Now, what are the examples? Biochemical reaction. It could be linear, like this. What is a good example? fermentation. Biochemical reaction could be a branch. What's a good example? Glycolysis. And the biochemical reaction could be a cycle. What's that? Citric acid cycle. All these details, except how we calculate the ATP, we're going to talk on Tuesday. Now today, we want to mention some basic knowledge you should already know. In order for bacteria to survive in their very short life, they need something magic. What is that? It's called an enzyme. Now, what's the function of the enzyme? The enzyme, they want you to have the energy required for the biochemical reaction is decreased. So, originally you may need that much energy. If you add the enzyme, the energy will be decreased. This is the difference when you add the enzyme. Of course, x-axis is the progress of the reaction, y-axis is delta G, is free available energy. Okay, that's what's the function of the enzyme. Now, how do we know the enzyme? And we already said, you have ASEs in them. So we talked about the catalase, we talk about the oxidase, we talk about the urease. You have an ASE, which means it's an enzyme. Now, what's the impacts for the enzyme? You already know. We don't need to be spending too much time talking about that. What are going to be the effective? Temperature is that. Most of the bacteria survive 35 degrees Celsius because the enzyme most active at that. Okay, what else? pH value, which means acid environment, alkaline environment is not going to favorable it to grow. Is that right? It's going to affect the enzyme. Now, enzyme also have a very specific. Usually we will say a group or specific group of the substrate is working on target enzyme. And lots of the time they also need a cofactor to do that. And most of the time the enzyme is, we call it a heat sensitive. Why? Because enzyme is a protein. But you know that not all the enzyme is heat sensitive. Some of them are heat resistant. Which one? If you know the PCR, we need a TAC enzyme. And TAC enzyme is heat resistant. Now why? Because they are isolated from bacillus. And the bacillus is thermophile. So that's another Nobel Prize for TAC enzyme to create it. It's from the bacillus. So these are the things we want to talk about the enzyme. Okay, now what are the components of the enzyme? We already said enzyme has a protein part. It also has a non-protein part. And the non-protein part has two different categories. Non-protein part we call the cofactor. If it is closely attached, we call it prosthetic group. If it is firmly, loosely attached, if it's loosely attached, and we call it a coenzyme. Coenzyme, lots of the time of vitamin. For example, vitamin B is necessary to create it folic acids. To biosynthesize folic acids or break down folic acids. So just want to let you know that's what happened. Now what is a good example for an enzyme? Lots of the time we also call it a halo enzyme. Is that right? Let's say we're going to teach you very quickly AEM polymerase. This is a very good halo enzyme. And they goes to closely attached, loosely attached parts, closely attached parts. There's lots of things we can talk about. Now regarding the enzyme, there is one thing it is very important that we want to talk and mention. We have the curve, x-axis, substrate concentration, y-axis, velocity, which means how fast the reaction could be. It's always like that. Because at the end of the day, it will become saturated. Okay, so how we do? We'll pick something right in the middle. This is Km value, that is 2, max. And what is Km values for? V max multiplied by substrate concentration. Then what is that? Sorry. That's V, that's Km add X. So what is Km said? Michaelis constant. What this means? Substrate concentration required for enzyme reacted at its half maximum speed. This is we call enzyme kinetics. In the biochemistry, you can teach this about 3 classes, how you're going to calculate it. And this curve also tells you, connected to the knowledge in the exam 1, why facilitate transport will go to stable status, because it's corresponding to the enzyme activity. Okay, so these are some of the basic knowledge. We just want to go over it. Now, how are we going to regulate the enzyme? How the enzyme will be working? Competitive. We could be competitive inhibit the activity of the enzyme. We mentioned sulfur drug. Okay, why they can do it? Because we needed to create folic acids. And we need a precursor called PABA, a beta-benzoic acid. What it looks like? That's beta-benzoic acids. And what's a sulfur drug looks like? Sulfur drug looks very similar to the structure. And sometimes it's exactly the same even. This is just a standard one. You can make it exactly the same. Majority of the parts are the same, only the head is different. So what happened? I have a substrate. Instead of PABA goes here, sulfur drug are going to go here. Once the sulfur drug goes here, what happened? You're not going to generate folic acids. The folic acids will be stopped. And the folic acid is an essential product for bacteria to survive. Of course, the bacteria can die. That is called a competitive inhibition. But you always remember, this is not going to touch anything for the cell wall structure and the cell membrane structure. So, it's going to be very effective? Probably not. We call this only inhibition. That's why sulfur drug only inhibits bacterial growth. Okay, that is something we want to talk. The second, we have some non-competitive reaction, which means the enzyme could be permanently not working with the substrate. For example, using heavy metal, like say mercury. That could be permanently damaged. But more important, lots of the time, what we do, we don't really work on the enzyme. We're going to attach something here for the substrate, and we change the substrate morphology, and let it be no longer fits. So the morphology may be changed like this. So it's not going to fit with the enzyme anymore. Remember enzyme and the substrate, what's the best model? Key and lock. So key lock model always the best one to explain. So I change the morphology of the substrate. Then the enzyme cannot work, cannot attach to the substrate. This is called allosteric inhibition. What are the good examples? We will talk about later. Lacto-opera. Okay, so this is what we have today. And they give you a very comprehensive overview of the knowledge in the biochemistry as an introduction. And these things in biochemistry class, you may take a couple of classes. We just have a one hour, more than an hour section to talk about that. So this is gives you some opening remarks like. But you can go ahead to study ATP. That will give you 5 points of the extra in the exam 3. Okay?